Today, I will explain complement fixation. First, I will talk about complement, then provide specific examples of complement fixation, followed by its biological role, the complement fixation test, CFT, and finally, a summary. What is complement? The discovery of complement began with the observation that antibodies alone were not sufficient to rise bacteria. An additional serum component was required. This component was thought to complement the action of immunity, and it came to be called complement. The main characteristics of complement are as follows. It loses its activity when heated at 56 degrees C for 30 minutes. Multiple types exist, which work cooperatively. It is inactive in the bloodstream. Most complement components are proteolytic enzymes. When complement is activated, it triggers a cascade in which additional complement components are sequentially activated. It ultimately results in the formation of pores in bacterial or cellular membranes. This series of reactions is called the component activation pathway. Depending on the initial trigger, there are three types of activation pathways: classical pathway, alternative pathway, Lectin pathway. Here, I will focus on the classical pathway. In the classical pathway, antibodies IgG or IgM bind to antigens on the membranes of invading bacteria or cells, forming immune complexes. Complement component 1, C1, then binds to these antibodies, leading to C1 activation. Activated C1 subsequently activates complement C4, followed by sequential activation of other complement components from C2 to C8. As a result, a complex of complement component 9, C9, is ultimately inserted into the cell membrane, forming pores in the bacteria or cells. Complement fixation refers to the phenomenon in which complement binds to antigen-antibody complexes and is consumed in the process. The activation of complement is not limited to rising antigens. It also plays an important role in host defense. For example, activated complement can attract neutrophils and activate macrophages and mast cells, thereby contributing to the immune response. Although the name complement may suggest a merely supportive function, complement actually plays a critical role in immune defense. Here, I will explain complement fixation test, CFT. CFT is a method for measuring antibody titers that exploit the properties of complement. In this test, antigen and complement are added to the test serum. As described earlier, complement is consumed when antigen-antibody binding occurs. Therefore, by determining whether complement remains after it has been added, the presence or absence of specific antibodies can be inferred. When complement acts on red blood cells, the membrane attack complex MAC is formed, leading to rupture of the red blood cells. This hemolysis can be assessed visually. Please look at the photograph. In test tubes that are red and transparent, hemolysis has occurred. This is because hemoglobin has leaked out following membrane disruption. In test tubes that are pure red and cloudy, hemolysis has been partially inhibited. In transparent test tubes, hemolysis has been completely inhibited. When antibodies against the antigen are present in the test serum, complement binds to the antigen-antibody complexes and is fixed. That's why complement is consumed. When antibodies are absent, complement acts on the red blood cells added to the test system, resulting in hemolysis. Based on this principle, the presence or absence of specific antibodies can be determined. The CFT is a specific antibody detection method that was widely used in the past. Although it is no longer commonly used today, understanding the principle of this test remains extremely important. Summarizing the key points of this presentation, complement is an essential component of the immune system and is activated through a cascade mechanism. Complement fixation refers to the phenomenon in which complement binds to antigen-antibody complexes and is consumed in the process. Complement plays an important role in host defense, not only by destroying pathogens, but also by recruiting and activating immune cells. The complement fixation test is a method for detecting antibodies that takes advantage of the properties of complement and allows for easy macroscopic interpretation.